So yeah, today I'll be talking to you about chromosomal inversions, kind of what they are, how they arise, how they play a role in adaptation, how they also might impact crop breeding, and then a little bit on what we're doing in the Senecio system to look at inversions. So the beginning of this talk will kind of be quite broad. So if you're an expert in inversions, you can just kind of sit back and relax. Okay. I'd first like to acknowledge the traditional owners upon the land on which we meet, which here in Brisbane are the Yugara and Turrbal people. We pay our respect to their ancestors and their descendants, both past, present, and emerging. And I'd also like to thank all the members of the OB lab, especially everybody involved in my work. Yes, and of course my supervisor, Daniel. Obviously none of this work would be possible without him. So a major goal that many of us have is that we want to kind of link the genotype to the phenotype and understand the genetic basis of traits. So here I have a little kind of diagram to show this. Well, on the bottom, we have the genotype, and on the top, we have the phenotype. So the genotypes on the left come together to produce one phenotype, and the genotypes on the right come together to produce kind of the other. And kind of many of us in the center, we want to do this. So we want to find out which genes underlie which phenotypes. So let's just say you're a researcher that wants to do this. So you might go out, you might find yourself kind of a nice study organism, which in this case is a nice tomato plant. Then you might want to look at the DNA of this plant to try and associate it to the phenotype. So you might get some nice leaf tissue, undertake DNA sequencing, and then kind of sequence the genome. And you'd typically maybe look at variation along the genome. So these are shown here as single nucleotide polymorphisms, or SNPs. And then you might want to try and associate these SNPs to particular phenotypes. Of course, it's kind of a lot more complex than that, but I'm just giving a very broad overview. Or you might be kind of interested in gene expression levels and how that underlies kind of particular phenotypes. So you might sample a few different tissue types, undertake RNA sequencing, and see what genes are being expressed. And you might do this under kind of specific treatments and experimental designs so that you can link gene expression to particular phenotypes. So these are the two main ways, kind of DNA and RNA, but the genetics kind of that underlies traits is not that simple. And there's kind of many other aspects of the genome itself which may contribute to trait variation. So let's kind of explore those a little bit further and look at the DNA of an individual a little bit closer. So here is one individual with their DNA, and here is another individual with their DNA. Of course, that's very simplified. These individuals, they could be from the same species or different species, but even though they're closely related, the kind of like the linear arrangement of their genes might not be the same. So let me explain that. So for instance, individual 1 might have one copy of gene 1, whereas this gene might be duplicated in individual 2, so where it has 2 copies of it. Individual 1 might have this tandem repeat, where a section of the genome is repeated, but in individual 2, this repeat might be of a different length. So this is copy number variation. Or there might be something called a translocation, where a segment of the DNA is in an entirely different position between the individuals. Or there might be something called a chromosomal inversion, where a segment of the DNA is actually flipped around and is in the opposite direction between the 2 individuals. So these things here, they're all types of structural variation, and they also contribute to the kind of the genetic architecture that underlies traits. So these are really kind of important to study. And I'm particularly interested in chromosomal inversions, which is obviously the focus of the talk today. So I'll now explain in a little bit more detail about kind of what chromosomal inversions are. So as I said before, chromosomal inversions arise when a segment of the DNA is kind of flipped or reversed around the other direction. So here is a schematic diagram of it. We have a chromosome and genes A to H from bottom to the top. And we can see that genes B to D get flipped around and inserted in the other direction. So this can happen in meiosis, where kind of double-stranded breaks in 2 regions of the genome can result in that segment of DNA being incorporated in the opposite direction. And it's often seen that inversions are kind of flanked by these repetitive regions of the genome or by regions of the genome with kind of these similar sequences. There are 2 kind of types of inversions, pericentric and paracentric. So pericentric inversions include the centromere, whereas paracentric inversions do not include the centromere. So now if we have like a diploid organism that has one chromosome with the ancestral collinear arrangement and one chromosome with the inversion, we refer to them as inversion heterozygotes. Okay. So what happens to inversion heterozygotes during meiosis? So don't worry about the details, I'll kind of step you through this, but effectively in the top two panels, we have a diploid individual with two homologous chromosomes. In A, we have representing a pericentric inversion, and B is a paracentric inversion. And so you can see that the order of the B and C genes in red are kind of reversed on the bottom chromosome. So this is an inversion heterozygote. So looking at the middle panel, during meiosis, when these homologous chromosomes come together in pair, when we have inversion heterozygotes, the region of the inversion kind of must form this loop for the chromosomes to properly pair together. So you can see that by that loop structure. Now, if there's a single crossover within that loop section, this produces gametes that either have kind of duplications or deletions of genes. So that's shown in that kind of bottom panels. You don't need to worry about too much about the details of those bottom panels. But the key point is here, if there is a crossing over event within the inversion, it results in unbalanced gametes, which likely die. So individuals that survive, so those that don't have a crossover, are those that don't have crossing over within the inversion. So effectively in inversion heterozygotes, recombination is drastically reduced in the inversion segment. There still can be a little bit of recombination within the inversion segment due to double crossovers, but yeah, that's not outlined here. So now we know that what chromosomal inversions are, kind of how they arise, and maybe how they impact recombination. Let's look at some scenarios of why they might kind of spread and persist in populations and species. Okay, so let's say we have two populations, population 1 and population 2. The individuals in population 1 are adapted to the green environment. So they're the genomes of them that you can see there. And as they adapt to this environment, they accumulate locally adaptive alleles, shown by those little dark green bits in their genome. The same thing happens for population 2, where the individuals are locally adapted to the blue environment and accumulate locally adaptive alleles. So in this case, natural selection is kind of driving divergence between the populations because they're adapting to different environments. But now let's say that this population is connected by gene flow. So gene flow is homogenizing the populations despite natural selection driving for divergence. So with gene flow, what happens is there's recombination between the two populations, which shuffles their genomes. So you can see their genomes are becoming shuffled here. So let's look at what is happening. So these two individuals here, they contain blue alleles that are only beneficial in the other blue environment. So it's likely that natural selection will remove them and those individuals might die. And then for the blue population, this individual with alleles or has alleles beneficial only in the green environment, so it also might die. But what happens to the individuals that contain locally adaptive alleles that are locally adaptive in kind of like both populations? So these are the ones that I've kind of circled here. So it is kind of very difficult for natural selection to remove them from the population because they have a little bit of some deleterious loci and some beneficial. So how does divergence in the face of gene flow occur despite this kind of genetic homogenization? So theory predicts that divergence can proceed in the presence of gene flow if there's a reduction in recombination. And we predict that this reduction in recombination is going to be selected for to surround only these locally adaptive alleles. So if we remember earlier in the talk, one way which recombination can be suppressed is with a chromosomal inversion. So let's explain that. So if we go back to the initial scenario, and if an inversion arises in one population that captures locally adaptive alleles, it will prevent recombination and breakdown of these locally adaptive alleles into the other population in the presence of gene flow. And so this inversion will kind of be selected for and spread in the population. So yes, they will decrease recombination, so they'll facilitate divergence. And there's kind of many studies in natural systems that have shown this, that inversions that capture locally adaptive alleles are favored during adaptation in the presence of gene flow. And a key point here is that for this inverted region, recombination is only suppressed between populations and not within each population. This is because for population 1, the inversion is kind of now fixed within the population, And then in inversion 2, that ancestral arrangement is fixed. So you get normal recombination within each population. This is quite useful because any maybe deleterious alleles that might accumulate within each population can kind of still be removed from that inverted segment, assuming they're not kind of too close to the beneficial alleles. Okay, so now to switch kind of gears a little bit, let's have a look at the potential impact of inversions in crop breeding. So how might chromosomal inversions kind of affect crop breeding? I'll just give you kind of a hypothetical example here. But say you have a particular crop represented here, and you're trying to increase the tillering of it, but you also want to increase the water efficiency of it as well. So here's the genome of your crop. And you might know that the gene or a gene kind of like of large effect controlling tillering resides here within the genome, whereas a gene of large effect controlling water efficiency is located here. Now, let's also say that these genes reside within a chromosomal inversion that's segregating within the population. So this means that there's no recombination between the genes. So they're kind of like linked together. And let's say that because of this inversion, kind of these traits might have a negative correlation where increasing the value of one trait reduces the value of the other because of this genetic linkage. And so because these genes are kind of trapped together in this segregating inversion, increasing tillering comes at a cost of water efficiency, and then increasing water efficiency comes at a cost of tillering, which is kind of not what you want. And this is, yeah, obviously because the genes are linked together. But if we are aware of which individuals were carrying the inversions, we might be able to break down this genetic correlation between these traits and disassociate them from each other. So to increase then tillering as well as water efficiency. So with knowledge of inversions, we might be able to make more kind of informed decisions of which individuals to select and breed together to create kind of better crop varieties. I don't really have expertise in crop breeding at all, but I'd be interested to hear from any of you guys if you kind of quantify and take into account these kinds of structural variation in crop breeding practices. Okay, so I just want to briefly overview how we actually detect inversions and kind of the methods that are out there to detect them. This is not an exhaustive list, but just kind of some of the main ways we can look at inversions. The more traditional approaches to detect inversions involved kind of like cytogenetics approaches. This is where like the heterochromatin in chromosomes were stained, or maybe the chromosomes were painted with different colored fluorescent bands, which could be used to reveal kind of large structural arrangements. Another approach to detect inversions is to construct genetic maps. And so this is where typically two individuals from a population are crossed together to produce kind of this large F2 mapping population. And in these approaches, inversions kind of appear as regions with either altered order of the genetic markers, or they might be kind of shown as kind of reduced genetic map distances due to the lack of recombination of inverted segments. And obviously with this approach, the ability to detect kind of like small inversions depends on the resolution of those kind of linkage maps. There are also quite a few genomic approaches that can be used to detect inversions. For instance, you can pick up signals of inversions with short read DNA sequencing data. So if your sequencing reads kind of overlap inversion breakpoints, or if you have kind of paired end sequencing data and the paired read kind of maps to a genomic location that is kind of a lot larger than the insert size of your library, you can detect it that way. Don't worry if I'm using too much terminology here for everyone. It's just, yeah, don't worry if you don't understand everything I say here. We can also detect inversions with long read sequencing data, such as HiFi or nanopore. It's great if the long reads can kind of span your inversions, or you might have used your long reads to construct multiple reference genomes or kind of a pan genome, and kind of aligning these reference genomes together will allow you to detect structural arrangements. You can also use something called optical mapping, where you have fluorescent tags, which can also kind of be optically mapped to your reference genome, which can also reveal inversions. Another approach is to use Hi-C technology, and this captures the 3D arrangement of DNA within a cell, which can be used to detect structural arrangements. Hi-C approaches are often used in combination with kind of population genetics approaches that look at linkage or recombination or kind of population structure along the genome. And because inversions have reduced kind of recombination, they will show this kind of large block of linkage disequilibrium and kind of like a different evolutionary history to the average of the genome. So these kind of high C and population genetics approaches is what a PhD student in the lab, Kath, is doing. Okay, so I think I have a few minutes left, but to conclude the talk, I just want to briefly overview kind of what we're doing in Senecio with inversions and kind of what we're asking with this type of data. Okay, so I guess most of you should be kind of somewhat familiar with the Senecio system that we work on. So it contains these multiple ecotypes that are distributed across Australia, and each ecotype kind of has a very distinctive morphology and occurs in a distinctive habitat. So there's a strong correlation between the environment and the phenotype. And a lot of our work, as you've probably heard, focuses on the dune and headland ecotypes. And these ecotypes are kind of interesting as they've evolved multiple independent times along the coast of Australia. So we have this kind of system of parallel evolution. Although there's kind of very little like contemporary gene flow between our populations, there was likely a lot of gene flow in the past, so around the time of the split of the populations. So this is kind of a really perfect system to look at the role of inversions during divergence in the face of gene flow. So this is exactly what Kath, a PhD student in the lab, is doing. So she's using a combination of whole genome sequencing and high-C data from a bunch of kind of dune and headland replicate populations, as well as some other ecotypes. So I'll show you some of the stuff that she's doing. Again, don't worry about the details of the figures on this slide. We can discuss it later if you're interested. But effectively, as a broad overview, to detect inversions, Kath is using a local PCA approach to first detect these kind of haploblocks in the genome. So regions of the genome that share similar evolutionary history that is different to the kind of average history of the genome. And these regions kind of indicate regions of reduced recombination. So they could be inversions. She's further confirming whether they might be inversions by looking at whether these regions of reduced recombination kind of cluster into 3 distinct genotypes, which is a typical signature of a chromosomal inversion. She's using high-C data to kind of confirm these structural arrangements in the genome. And then she's also looking for signatures of selection in the genome. So this is things like FST, DXY, nucleotide diversity. And she's asking whether these inverted regions show signatures of selection. So are they kind of under adaptation in the system? And so with this, we're finding some really good candidate chromosomal inversions, also ones that correspond to genome-wide association studies of adaptive traits we've done in the system. So we have some nice inversions that are corresponding to regions of the genome that might underlie gravitropism, as you've heard about in the system. And then we'll kind of complement all of this work with the Senecio pangenome that we are creating to kind of confirm these inversions with another line of evidence. So finally, we can kind of answer many questions with this, such as, have chromosomal inversions facilitated adaptation? Do inversions capture locally adaptive alleles in the system? Have the same inversions arisen multiple times across our replicate kind of dune and headland populations? If not, do these inversions kind of capture genes with similar functions across our replicate populations? And finally, we'll also be looking at kind of, do inversions actually reduce recombination rates? So this will be combining some data from some other PhD students in the lab. So overall, I think it's really important to kind of look at this structural variation in the genome. So not only look at DNA and RNA, but kind of how the genome is kind of, structured to kind of get a complete picture of the genetic basis of traits and to try and link that genotype to the phenotype. So I think that's pretty much all. I'd just like to thank everybody in the lab again. Thank you.